So let's review where we are. We've analyzed equilibrium reactions in chemistry, and we've come to realize that there's this delicate balance between reactants and products. And at equilibrium, the relative concentration, the relative pressures are the same, but there's rapid interchange between those reactants and products. We've also come to realize how important Gibbs free energy is in the analysis of the landscape over which these reactions occur. And we've come to realize that living organisms use chemical equilibrium because they can control the relative concentrations of reactants and products by subtly shifting the conditions under which this chemical equilibrium exists. And that brings us to our issue of stress. We can apply stress to a chemical equilibrium, and we can see how it adjusts. And we have done this with stress applied as a pressure change, and that pressure change results from a change in volume. And we looked under conditions where we have more molecules on the left-hand side than the right-hand side. We've looked at conditions where we have an equal number of molecules on the left-hand side and the right-hand side. And we've looked at the final case where we have more molecules on the right-hand side of the reaction than the left-hand side. And we've come to learn how that chemical balance adjusts to moderate the implied stress. And in this case, the stress is applied by decreasing volume, which gives us the increase in pressure. We've also analyzed the response for the injection of a molecular reagent into this system that was otherwise at equilibrium. And we've watched the equilibrium shift in response to that implied stress through the increase of one of the species. And the reactions respond by adjusting to moderate the overall impact of that applied stress. And finally, we've investigated this very important property of temperature. And when we change the temperature of the system, we actually change the equilibrium constant. So we have written our equilibrium constant K, and we referred to it as a constant, which it certainly is, except when temperature is changed. So we have to put a little quotation mark around that and realize that the applied stress by, for example, increasing temperature favors a reaction with a higher barrier, and so the system adjusts accordingly. And we've noted a consistent picture here that in each case the system perturbed from equilibrium by the stress will shift its equilibrium position to relieve the applied stress. That's a qualitative conclusion we've come to, but it turns out it's extremely important because when we look at a chemical equilibrium process, the first thing we need to do is analyze the landscape over which that reaction takes place, which is the Gibbs free energy surface, and we have to analyze the response to various external stresses applied. Well, this brings us to the principle of Le Châtelier, and Le Châtelier was a very famous French chemist in the 19th century. And his statement of how to interpret the response of a system to an external stress is to say that if a chemical system at equilibrium, so it's very important that the system has achieved equilibrium, and if that equilibrium experiences a change in concentration, temperature, volume, or total pressure, then the equilibrium following the application of the stress shifts to partially counteract the imposed change. And that principle of Le Châtelier has survived through all of the interpretation of equilibrium chemistry, and it provides the qualitative way for looking at the response of a system to an external stress. The next thing we focused on, and we need to examine with increasing quantitative analysis, is this crucial relationship between Gibbs free energy, the surface of Gibbs free energy, and the value of Q and K. And we noted that if we're looking at our favorite reaction, which is A plus B going to C plus D, which includes the stoichiometric coefficients, when we start with pure reactants, delta G is less than 0, driving this reaction spontaneously from reactants to products. Equally true, if we start with pure products, that equilibrium reaction will be driven again by Gibbs free energy being less than 0 and a spontaneous pathward down to the bottom of that landscape of Gibbs free energy until we reach the point where Gibbs free energy is equal to 0, and at that point there's no motive, no chemical motive to adjust the ratio of the concentrations, and at that point we have reached equilibrium. Now, when we apply a stress, then we have a calculation to do, and the calculation is to determine the value of Q, the reaction quotient, immediately following that stress. And so we remember that the functional form of Q is identical to the functional form of the equilibrium constant, except that we can calculate Q anywhere along this reaction coordinate. So Q is then equal to the concentration of C raised to its stoichiometric coefficient, the same for the other product D, and identically for reagents according to the law of mass action. So when that stress is applied, we calculate Q, and for example, if Q is less than the equilibrium constant, in this case it's our equilibrium constant written in terms of concentration, then we know immediately that Q sits to this side of the equilibrium point, which is to say when Q is less than the equilibrium constant, we know immediately that the system is going to inexorably drive back to the equilibrium point, which means it's going to shift from left to right. In exactly the same vein, if Q is greater than the equilibrium constant, then the system is inexorably going to drive from right to left, and that shift then carries us back to the point where delta G is equal to 0, because the system is always trying to reach a condition in which the change in Gibbs free energy is equal to 0.